You might need to use small volumes of liquid that fit in Eppendorf tubes. These tubes should be contained in a previously autoclaved container that has been introduced in a clean manner, after spraying ethanol on a tissue and wiping the surface of the container. To take a tube out, do that inside the hood. Tips are used for pipetting small volumes of liquid and should also be in previously autoclaved containers and introduced in a clean manner in the hood. The lids should be closed at all times where you are not taking a tip out. Containers for Eppendorf tubes and tips in use are usually kept in the hood at all times. Eppendorf tubes are taken out one at a time and placed on the appropriate holder. Make sure that you do not touch the opening of the tube with your glove. When done with the container, close the lid. Remember to label the tube on its side with short information on its future content. Take a tip from the box that you should close immediately afterward. Take the amount of medium needed by pipetting from the upper portion of the liquid. Pipette carefully up and down a few times. If the medium contains something that needs to be mixed, it could be cells, for instance. Place the lid back on the tube and add the medium inside the Eppendorf tube. The tip should not touch the walls of the tube and medium should be added slowly in order to avoid for the liquid to splash out. Close the lid of the tube immediately. If you need to take a small volume from the Eppendorf tube, for instance when you need to count the cells from an aliquot, open the lid by pushing from under it. Introduce the pipette tip slightly inside the volume of liquid. Stay in the top third part of the liquid, pipette up and down slowly to mix, if there are cells for instance, and take the needed volume out. Make sure that the tip does not touch the inside wall of the Eppendorf tube when taking the tip out. It is important to take very small volumes from the upper part of the liquid in order to keep the volume exact. Taking the liquid from the bottom of the tube might result in liquid sticking to the outside surface of the tip, which should then add an unwanted amount of liquid to the one you initially wished to take.